Well, it is 4 p.m., so I just started the recording, and I think most people here have met Dr. Streicher before, but I just like to introduce him real quick for those of you who haven't. Dr. Streicher is a professor in the Department of Pharmacology and also a professor of neuroscience at the U of A. And most of his research is about GPCR signal transduction. And a large contingent of that is with regards to opioid receptors and cannabinoid receptors, and essentially the networks in which they work together or interrelate. And he's also a member of the Comprehensive Center for Pain and Addiction, which the Largent Milnes Lab is also a member of. And he is a member of my doctoral committee. And so, Dr. Stryker, we're excited to hear from you.
Thanks, Sophia. I really appreciate the invitation, and I actually really appreciate what you all are doing. So, like, we haven't had a lot of students show that kind of initiative, and coming together on their own to do something like this. So that's pretty cool. I think you all should be proud of yourselves for having that drive. I'm just going to move that over there, so everyone can see this normally. Yeah.
Mhm.
Okay. So today I want to talk a little bit about our work with terpenes. So we actually do a lot of different things. So like Carolyn was mentioning Adrian a second ago. So like Adrian worked on our GPR project, orphan receptors. We did a lot of stuff on opioids, as was mentioned a second ago, and signal transduction. But we've also been doing some work on cannabinoids, in particular terpenes. And so this work came from one of my former students, Justin Levine. He graduated years ago, but he started us on this path. He just got interested in it, said, "Hey, would like to study these." We bought a few; they're pretty cheap. Tested them out, and then it was kind of off to the races since to characterize what they do behaviorally and mechanistically, and then also now branching into therapeutics, looking at chronic pain and reward as well as I'll talk about today, but even other areas too. So my student Emily has gotten really interested in depression and would like to try some of these in a depression model. So they're interesting molecules, and there's a lot that can potentially be done with them. So what the hell is a terpene, first of all? So this is everyone's favorite plant, which is the gets all the attention and excitement. By the way, I published a lot of papers in my career. The first paper that we published on these terpenes by far was like the most public interest and excitement of any of them. Like just like it was not in a high impact journal, like it was a fight to get it published, but like it really kind of this topic kind of captures the public imagination. So I guess if you want to get a lot of attention, go into cannabis, I suppose is the lesson from that. But the number of members of the general public, companies that are in this space and want to collaborate has just been huge. So it's just like all over the place and been a lot of interest and excitement. But anyway, this is this plant that everyone knows and loves, but of course the different components of the plant are not all the same. And in particular, when you focus on the area near the top of the plant, you had these little structures here, like these little kind of dewdrop looking fellas. These are called trichromes. This is where the area of heaviest concentration of different chemicals and ligands in the plant. And many people have studied these over the years. And of course, we've studied the compounds that we all know about, know and love, cannabinoids, delta 9, THC, tetrahydrocannabinol, as well as cannabidiol. Those are the ones that get most of the headlines. And to be fair, encompass the majority of the actual amount of drug in the plant, at least for most strains. But there's a lot of other stuff too. So first of all, there's a lot of cannabinoids. I don't even know what the number's up to now. There's got to be like 20 or so that have been described at least. And not all of them have a lot of research or information done on them. So we're talking about drugs you may or may not have heard about, like THCP, CBN, CBDA, like THCA, like all these different variations on the cannabinoids. And there's also a lot of non-cannabinoids, and terpenes are one of these groups. So beta-myrcene, beta-caryophyllene, humulene, little, you may have heard of some of these words before. And the cannabinoids get the attention, but these other molecules, there's no reason to think that they might not have their own activity. And in fact, they do. And there's also been a lot of interest in this concept of the entourage effect, the idea that the different components of the plant might work together in some way or add to each other in some way in order to give an effect that's different from the purified component alone. So some of these backgrounds and ideas drove kind of our interest in studying these terpene molecules. And so when Justin started, back in what, 2019 or so, I guess, was when this first study would have started, 2018, maybe a little bit. These are the first five that we selected to study: beta-pinene, linalool, geraniol, alpha-humulene, and beta-caryophyllene. And as you can see here, these are not just five different pictures of cannabis. These are different plants. So these terpenes are not unique to cannabis, but they are heavily expressed in cannabis, and a lot of the interest in them has derived from the cannabis field, but they're found in other things. So beta-pinene found in pine trees, linalool found in lavender, geraniol in roses, and so on, humulene in hops, beta-caryophyllene in peppers and cloves. So a lot of things you may be familiar with. So it's like you have experience with these molecules, even if you've never heard the name before, because you've smelled these smells, you've tasted these tastes from these different plants. But for a long time, of course, everyone thought that's all they were doing, just taste and aroma or whatever. But they do have therapeutic properties. These have become obvious over time, are more obvious over time, have been tested in different studies. And so we selected these five based on, first of all, that the fact that most of them were expressed at least a reasonable level in the cannabis plant. And there was some indication from the literature that they had some sort of therapeutic benefit, analgesic, anti-inflammatory, antimicrobial, and so on. Although I would characterize this literature as wide but shallow. So there's a lot of studies out there. The vast majority of them, at least until recently, did not go into any depth. I don't want to slam everyone or say no one did. That's a little bit too far. But in general, and that is starting to change recently. So there's our work, of course, but there's others too. There's another recent study from Kathy Cahill, and others in the field have started to dig into these in more detail. So I guess we're not alone. But anyway, we started with these five, and we wanted to look at them in more detail to figure out what they're doing and whether or not they might be combining with cannabinoids or other things in a different way. And so you can read our initial paper in 2021. What I want to focus on, though, is the work that came after that. So we did the initial characterization, and then, of course, our lab is very interested in pain. And so we said, hey, could these be effective in relieving chronic pain, pathological pain, and other types of pain that we would like to treat? And so we tested that. This work was relatively recently published in 2024. Abby was sort of the lead author on most of that work. Abby Schwartz from my lab just graduated last year. And so when we inject these terpenes into, in this case, chemotherapy-induced peripheral neuropathy mice, they produce a pretty solid anti-nociceptive effect. This is, as a control, a dose response of a cannabinoid called Win55212. And overall, when you look at the area under the curve, the terpenes are producing a solid effect that in most cases is pretty comparable, if not statistically the same, as that produced by both cannabinoid and morphine positive control. And so right off the bat, we're like, hey, that's a pretty solid amount of pain relief. On the other hand, we know from the literature, at least suspect from the literature, that there really aren't a lot of side effects to these things. Beta-caryophyllene in particular, down here, has been approved for general human use, not for therapeutic use, but as just as a sort of a general purpose use, as generally recognized as safe by the FDA. So we're thinking, hey, there's not a lot of side effects or other problems with these. They look to be pretty effective in some of these pain models. So maybe there's something here. Maybe we can dig in, figure out if they actually could be good therapeutics, and figure out a mechanism, because no one had really identified any kind of significant mechanism for how these could be pain relieving. And so we dug into detail. Overall, I guess this is sort of a summary of what we found in that 2024 paper, since I don't have the time or the desire to go over all of that in detail. And we found some things which suggest that they might be pretty good therapeutics. They have pretty high efficacy, as you showed, sort of similar to opioid and cannabinoid. We showed, I'm not showing this data, but they synergize with opioids. So when you combine a low dose of opioid with a low dose of terpene, you produce more pain relief than either alone, which might allow you to basically engage in a dose reduction strategy. So you give less opioid, therefore you have less opioid side effects, but the pain relief is maintained or even improved. We also showed that there was no reward liability. We tested this quite exhaustively. There's definitely no addiction liability. Some of them are aversive, but overall, certainly not addictive. That's good. There was one small deficit. We did discover that they do show tolerance in a similar way to opioid, but that's no worse than current treatments. And then lastly, as I mentioned, these are generally recognized as safe and also commercially available. So I've had people contact me and saying, "Hey, Dr. Streicher, can I get into your clinical study?" I'm like, "Well, my friend, you can do better than that. You can actually go and buy this stuff and actually just try it out and see if it works for you." I mean, it's an unregulated space, so you got to be careful. But there are products that are available. This one in the middle, I work with this company to develop and test their product in mice as well as in humans. And so there are some products out there that can be tested. So this is a nice thing in terms of a therapeutic. We don't have to go through 10 years of study, get a new chemical entity IND and all this and all the work and expense and everything that goes into that. So this is available. And so they might be good therapeutics, but I did want to focus a bit on more on their mechanism, which leads into some of my more recent projects that haven't been published. And in particular, we wanted to know what was the mechanism in pathological pain. Our first thought was cannabinoid receptor 1, because everyone thinks about terpenes. Usually the first thing they'd say, "Hey, is this a cannabinoid?" And it's worse. So the first thing, Abby ran that with a rimonabant, no difference with any of our rimonabant treatments. Is clearly not working through the cannabinoid 1 receptor in this pain model, at least. And we're kind of scratch our heads. Well, what could it be? We went back to our 2021 paper where, for various reasons, we had tried an adenosine A2a receptor antagonist called istradifylline that did block some of the effects, at least in some of the tetrad activities of these terpenes. So we thought, okay, let's try istradifylline. Let's try the adenosine A2a receptor. It might be that. Luckily for us, it was. So here now in two different pain models, not CIPN, the postoperative paw incision, as well as a fibromyalgia model to mix things up a little bit, we find that the A2AR antagonist istradifylline completely blocks this effect. So here you have your terpene effect. You see it's pretty strong, producing pretty good antinociception, and that is just completely flatlined by the istradifylline treatment, right? So completely blocks the effect. This is the quantitation. And this, as well as other studies, this is just a sort of a flavor of that, really pointed the way not towards the cannabinoid receptor, but the adenosine A2A receptor. And that has been a lot of our focus in this work ever since we kind of initially discovered that. Not really in the cannabinoid receptor, to our initial surprise. And of course, an antagonist, there could be, as you're all or many of you are pharmacologists, you know, could be off-target effects, could be other things going on. So we also wanted to use another method. We used a CRISPR approach. So we take this all-in-one DNA CRISPR, we can complex this as transfection reagent, transfect it into the spinal cord or even the brain, and get tissue-wide knockdown in adult wild-type mice. This is really convenient. Like, you don't have to make a knockout mouse or whatever, which takes ages to make. Pretty quick, relatively easy. And we tested that in these terpenes. Beta pinene is not shown for reasons I don't want to get into. It was a little bit complicated. And it's solid effect in 3 out of these 4, and maybe a medium-sized effect in geraniol, confirming the initial finding. And so targeting or sort of pointing the finger at adenosine A2A receptors, and also in this particular case, pointing to the spinal cord, since these were injected intrathecally, which will be important here in a few minutes, right? And so we kind of start wide. We're starting to narrow down adenosine A2A receptor, at least a site of action, maybe not the site of action, but a site of action in the spinal cord, and we're starting to narrow this down. Sorry, let me sound the way. All right, so adenosine A2A receptor is involved, but these experiments don't demonstrate that how the terpene is interacting with the adenosine A2A receptor. So the initial hypothesis most people would suggest is that, oh, it's an agonist at the receptor. Maybe it is, but maybe it activates something else, and adenosine is sort of in the cascade downstream or something along those lines. And so we wanted to test that using in vitro models by testing if the terpenes could activate the adenosine A2A receptor directly. And so we started with just an in vitro cyclic AMP assay. The adenosine A2A receptor is G alpha S coupled, so it directly stimulates the production of cyclic AMP. NECA is the positive control agonist. We get a strong effect. And then all of the terpenes produce this sort of solid partial agonist type activity in this assay. And so all right, great. So we think it could be at least interacting directly with the A2A in the spinal cord to produce this antinociceptive effect. Here's some evidence, at least, that it can interact through the adenosine A2A receptor. Where things got a little bit complicated, and this wasn't a total surprise to us for based on our earlier studies, is when we did a binding assay. And so we're combining the terpene, in this case, with this tritiated ZM compound, which is a known ligand of the A2A receptor, and we get no competition with any of these terpenes, even up to a very high concentration. And so we're like, wait a minute, all right, we stimulate, and in cells even, we stimulate, we can get sort of agonist-like activity. It doesn't seem to be an orthosteric agonist. It's not competing with the at the orthosteric pocket. Could it be an allosteric agonist? Could be activating at some other site. And we still don't know the full answer to that question, but one experiment that we did suggests that it is. So we did this type of assay called a plasmon waveguide resonance. This is like, I mean, I don't fully understand it myself, but basically you have your receptor protein. It's reconstituted in an artificial membrane, suspended in this like assay chamber. You're basically bouncing a laser off of it, and the instrument is so sensitive that you can detect small shifts in the reflection of that laser depending upon the conformation of the protein. And so as some ligand binds to the protein and changes its conformation, you can actually detect that shift. And when we tested all the terpenes, dose or concentration response here, you can see we get a very nice concentration response curve. We get this angle shift suggesting that these actually are interacting with the receptors. These are purified receptors now. And then down here is just the lipid, the bilayer, the artificial bilayer, no response at all. So this is a pretty good indication that the terpenes are interacting with the receptor, at least can interact with the receptor. Doesn't seem to be orthosteric, but it probably is allosteric, acting at some other site. And that's something we're going to chase down, hopefully in the future, to figure out exactly how these are sort of interacting. But overall, through these studies, this is kind of a gloss on them. Overall, what this suggested as a mechanism is that we have the adenosine A2A receptor in the spinal cord. The terpenes are activating that to produce some kind of pain relief in these pathological pain models. And then this does actually combine with opioids, but not cannabinoids, to produce enhanced effects. And this is basically where we were at in 2024 with the publication of this paper. And this does provide some important information, right? So it suggests a mechanism of action. It suggests location. These are good things. It does suggest the potential of these ligands for therapy, that we could potentially give these to patients and achieve pain relief. It has good efficacy, side effects, blah, 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 blah. All that's great, but of course there are a lot of unanswered questions, and that's really what we've been chasing since then. So my current student Jerry, and as well as Hager and now Emily, are chasing down the additional questions that come after that, and forging into new areas, which is kind of what I really want to talk about. And so the first one is a natural extension. So we say it's the adenosine A2A receptor. We say it's in the spinal cord. We've started narrowing down. Well, we want to narrow down even further, right? We actually want to identify a circuit, an actual set of or a set of neurons in the spinal cord that could actually achieve this pain reduction effect. And we would be the first, by accomplishing that, we would be the first to do so. So no one has identified any circuit mechanism of action for how these ligands work. And so Jerry forged ahead. We had a lot of negative results trying to co-localize the A2AR with these different molecules until we hit upon PAX2. So what is PAX2? It is a marker for inhibitory interneurons in the spinal cord. And so not just one type. So this can include GABA, this can include glycine, but generally speaking, this is inhibitory interneurons. And when we did the co-localization, we had the adenosine A2A receptor in green. We get a really beautiful stain. It's actually really, really nice. Like it's one of the easiest targets we've ever had to do for immunohistochemistry. But then when we co-localize with PAX2, we're getting solid co-localization. About 60% or so of the A2A population co-localizes with the PAX2. So that doesn't mean that the terpenes are acting through these, but that's an initial strong sort of premise, right? That the adenosine A2A receptor is located in these interneurons, these inhibitory interneurons. We know that the inhibitory interneurons can block ascending pain input, and we know that the A2AR is a stimulatory receptor, which would make sense, right? You're stimulating these inhibitory neurons, providing more inhibition, thus reducing pain. That was sort of our thought when we hit upon this PAX2 co-localization. But of course, this is just an initial correlation. Much more has to be done. And so the next thing Jerry did was targeted the A2AR in PAX2 cells. And we adapted our approach. This was first published by us in 2023 by Catherine, former student in the lab. What she figured out, what Jerry took advantage of, is we had this initial CRISPR DNA construct I told you about earlier. That initial construct had a cytomegalovirus promoter that will non-specifically and strongly drive the Cas9 expression. So basically, any cell that this gets into, it's going to knock down whatever your target might be. You can make whatever target you want. In this case, our target is the A2AR, right? So we want to knock down A2AR. However, we don't have to use this promoter. And so Catherine used the CGRP, the Calco promoter. We've also done the AIF1, the slash IBA1 microglial promoter, and now in this case, the PAX2 promoter to drive selective expression of the Cas9 just in particular cell types. And so this is some data from Catherine's paper in 2023. In the negative control, you see that colocalization with CGRP in this case. With the selective CRISPR, that is selectively lost. You still have other signal, but you don't have that yellow colocalized signal anymore. So it's knocking it out in selective cells. So we took advantage of that. Jerry swaps in the PAX2 promoter here. We have the A2AR guide RNA, and so now we should get knocked down just in the PAX2 cells. And when he did that, we get this very nice complete loss of terpene antinociception. So this is really great, right? So this now suggests not only is the receptor in this particular cell type, but now when you knock it out in that cell type, you lose the antinociception from the terpenes. And so this is now connecting them together more fully. Of course, there's a lot to do, but at this point, we have enough to sort of suggest a model. And so you have this ascending input that comes in through the dorsal root ganglion into the ascending primary nociceptors. That pain signal is transmitted to the second order neurons in the spinal dorsal horn by neurotransmitters like glutamate, substance P, CGRP, and so on. And what we think is happening is we have this PAX2 interneuron that's releasing GABA onto these pain neurons, reducing that signal. And the A2A receptors on these PAX2s are going to be activated by terpenes to sort of block this effect and to block pain. And so that's the current hypothesis. Jerry's now trying to do the additional sort of testing and experiments to fully confirm all the elements, to measure the GABA release, to measure the activity of these second order neurons, and so on, to fully establish this model and to get it published so that he can graduate in the spring. We have a little bit of data for that in terms of staining for FOS. And so for those of you who don't know, FOS is an immediate early gene. It is a marker of neuronal activity. And as you can see here in the sham/vehicle treated animals, there's very little FOS activity in the spinal cord. However, when they are in this pain state, chemotherapy-induced peripheral neuropathy, in this case, you see now these neurons light up. So these are neurons that are receiving pain input from the periphery. Now they're activated. The FOS is measurable. And this is one marker of pain activity being transmitted into the central nervous system. However, when we treat with geraniol, we lose most of that, right? So that's kind of quantitated here as number of cells. You lose not all, but most of that signal. It looks much more like the sham. And so that is now some indication that you treat with the geraniol, the terpene, you're losing this pain signal here. We still, of course, need to do the colocalization and show that it's these specific cells which are losing this activity. Jerry's also actively working on the GABA measurements, and then we're also going to do some dreads, express dreads in these cells to fully tie together this story, but that's well in progress. And I think he'll be probably wrapping that up in the next few months to sort of demonstrate this circuit. Any questions on this, by the way, before I shift gears a little bit?
I do actually have one comment, which is that I did not know about PAX2, and these interneurons also happen to have GQ-coupled oxytocin receptors, which you might find interesting. Yes.
Yes, that is interesting. Of course, to your project, but also we're working with PULT on oxytocin ligand that can get into the central nervous system. So that could be a mechanism by which those are produce pain relief, which we show they can. That is interesting. Thank you.
Actually, I have a question.
Yeah.
Why are you using geraniol instead of one of those other three terpenes that look like they had 100% efficacy? This one had like 50%, right?
No, geraniol was one of the strongest. So if you go, yeah. So geraniol is here in purple. So this one technically is, I mean, it's not statistically different, but it is kind of one of the more effective ones. And ultimately, we haven't really discovered very many differences between these five in terms of how they activate the receptor and produce pain relief. And we kind of had to pick one. And it's like, well, geraniol is a good one. It produces a strong effect. No aversive properties in the CPP assay. So that was kind of the reason that we selected that one.
Okay. On the next slide, was it acting through more than one receptor though?
Oh, yeah, the you're talking about this one, right? Yeah, so with the incomplete knockdown with the CI, or sorry, with the CRISPR, that is true. But on the other hand, when we do the geraniol with istradifylline, the effect is full. And so there could be other sites of action, other activities or other sites could be contributing to it. But so maybe there's, I don't know, adenosine A2a receptors in the periphery or in the brain that are contributing to this effect that's not captured fully in this knockdown. But overall, we can fully block the efficacy by the use of a A2AR antagonist.
I see. Okay.
Anything else? All right. All right, so this one's really cool, though. I've been excited about this one. So being able to actually identify a circuit for this one has been a lot of fun. I'm kind of becoming a real neuroscientist because like actually figuring out circuits. We haven't done much of that before. We're getting more into it, though. All right, so circuit. So he's working on that. It's should be done soon. Another thing that Jerry's been working on, which we found kind of interesting, is that initially we found that the terpenes are highly effective in relieving pathological pain, but not at all effective in sort of nociceptive or uninjured pain states. And an example of that is shown here, right? So contrast, these sort of results, right? So this very strong efficacy, say in CIPN, comparable to morphine. You contrast that with their effect in a nociceptive pain. So we here have three different pain modalities: thermal heat, thermal cold, and mechanical. In all cases, the morphine or oxycodone positive control produces the expected response, but the terpene doesn't. Right, terpene here is really producing no response at all, where it is producing very strong effects in the pathological pain, paw incision fibromyalgia, CIPN, and LPS-induced inflammatory pain, and so on. And so that got us wondering why. And one of the things we happened to notice when Jerry was doing his staining, so initially when he was staining for the A2AR, he always used CIPN animals. I said, all right, Jerry, we got to throw a sham in there as a control. They're going to want to see that to see if there's any difference. And he ran the shams, and there's very little in the spinal cord, right? So pretty low expression. And then it really just lights up with the CIPN. We've done this now, repeated this and quantified this. We're getting like a threefold or so stimulation of A2AR in these pathological pain states that's not present in the sham. And that got us to thinking maybe this upregulation is why the terpenes are not effective here where their molecular target is low, but they are effective in CIPN and other pathological pain where this is upregulated. So Jerry is now sort of wrapping up the behavioral. So we've done the same assays here in the uninjured mice as well as the CIPN. Those CIPNs are still in progress, so I'm not showing those. But we have all that, sort of matched behavioral data. We know this is upregulated, and then he's going to wrap that up with some like dreads and selective overexpression type experiments to sort of connect those two things together and demonstrate that this upregulation could be indeed responsible for this difference between the different pain states. But so scientifically, that's interesting, but also therapeutically, this is kind of beneficial if you think about it, right? And so one of the reasons that a drug like, say, morphine is more desirable than a drug like lidocaine for general use is that it doesn't completely block, or at least doesn't block that much, normal pain perception. Because when you lose normal pain perception, that's a good way to get yourself injured normally, right? You put your hand on the hot stove, you don't realize it's there. Most of the time, we prefer to preserve that kind of pain relief if we can. And here, the terpenes seem to be kind of selective for the pathological pain, which honestly would be ideal, right? That they're active in the pain that we care about and not active in the pain we want to preserve. So that project isn't as advanced as this other one, but that is also ongoing. It's kind of an interesting thing. And then another project area we've been pursuing, kind of in progress that I wanted to share with you, is the effect of terpenes on reward. And so this is something that Abby kind of just initially, I mean, most of her thesis or dissertation was on pain, but she's always been addiction, or always was and is interested in reward and addiction. And so wanted to try some of these things in some sort of reward modulation. She made some advances in that before she graduated. We're now continuing that work. But this question started with this initial CPP observation. So I mentioned to you that none of the terpenes were rewarding, which is true. Here's your morphine producing that expected positive reward association. Several of the terpenes were neutral, geraniol, linalool, definitely. Statistically, pinene is neutral, but that's looking a little twitchy, but whatever. Like you get some ones here that are neutral. And that, of course, is ideal as a therapeutic, right? Neither rewarding nor aversive. Like that's what you want for a therapeutic. And then initially, we found that these two, alpha-humulene, beta-caryophyllene, showed aversion in this assay. So initially, like the first sort of reaction was, oh, like that, okay, that's cool. We'll just not use these for therapeutics. They would cause aversion. Let's use these instead. Maybe that's true as a standalone, but then we got to thinking in a little bit more detail. Like maybe we can use this aversion because we did show that these drugs combine with opioid to produce enhanced antinociception. Maybe this aversion will cancel out the reward that morphine produces while still producing enhanced pain relief, right? So as a way to sort of block that rewarding. And in fact, no one in the literature has done this for opioids, but they have done it for other drugs. And so that sort of showed some premise that this might work. And so Abby started testing this in opioids. And the first thing, of course, was just to combine the drugs together in a CPP assay, condition-placed preference. And so again, here's your morphine producing that expected positive deflection. And when you combine beta-caryophyllene, now that very nicely brings you right here into the middle, that neutral zone. And this would suggest that when you combine these two drugs together, morphine and BCP, you're going to have neither reward nor aversion, which is exactly what you want. You've sort of taken away the negative properties potentially of both drugs. Taken away the reward of morphine, the aversion of BCP, they meet here in the middle, while still having the advanced pain relief. Right? And so that could suggest a therapeutic approach of combining these together, maybe some sort of combinatorial therapy. We're pursuing that project area in terms of therapeutics. Also working with a former U of A Department of Pharmacology grad, Lauren Slosky. She came out of Todd's lab. She's now at Minnesota. She's going to help us do IVSA, intravenous self-administration, and really show the effect of the BCP on sort of addictive, true like addiction behavior. But while we pursue those therapeutic aspects, of course, we want to know mechanism. Like, how is this actually occurring? And again, there's very little on this in the literature. And so we started looking at some of the different aspects of the reward circuit to determine if these could be BCP, beta-caryophyllene could be modulating that. And so one of the first things we did was look at tyrosine hydroxylase staining. We tried to do dopamine microdialysis, but it turns out that's extremely difficult to do in mice. So maybe we'll do that someday in rats. But so we had to settle for staining for tyrosine hydroxylase. And so these are the blown out or zoomed out sections. Here you can sort of vaguely see the ventral tegmental area. It's much brighter in the morphine conditioned animals. You get this big upregulation of tyrosine hydroxylase in the VTA. Of course, these neurons project to the nucleus accumbens and release dopamine in response to opioids as well as other drugs of abuse. You get these nice bright cell bodies here now. This TH is upregulated. And then interestingly, we found when you combine these, BCP and morphine, just like BCP blocks this reward effect, it very nicely blocks the upregulation of the tyrosine hydroxylase. That is shown very cleanly here in this quantitation. The morphine, you get that upregulation. BCP, and in this case, alpha-humulene alone, don't produce any effect on their own. And then they block that upregulation by morphine very nicely. So this is now starting to show, indeed, so it behaviorally it blocks the effect, but also on the molecular mechanistic level, it can clearly interact with at least some parts of the reward circuit. But again, that's not enough. We want to dig into more detail. And so the initial experiments or the experiments in pain have linked the terpenes in pain relief to the adenosine A2a receptor. And so naturally that was our first thought when testing for pain, testing for reward. Could it be through the A2a? And the literature, of course, shows that the A2a can modulate reward and addictive behavior. And so these experiments are still in process, but we did complete one experiment. So we did our CRISPR approach, not in the spinal cord this time, but in the brain, and knocks down the adenosine A2a receptor. This is the nucleus accumbens. And so initially here with the negative control, BCP alone produces aversion. You combine them together, that aversion, or sorry, not combine them together, but this BCP alone produces the aversion. You combine that with A2A knockdown. Now that aversion is gone. So the sort of the aversion modulation or reward modulation effects of BCP are mediated through the adenosine A2A receptor, at least here. And then, of course, we're now in the middle of doing the experiment where we combine this with morphine to show that BCP blocks the reward effect of morphine, that we can reverse that effect with A2A knockdown. That is literally in process. We've done like 3 of the cohorts or something like that, and hope to have that soon to tie that together. And so this is sort of early stages in terms of figuring out mechanism, but we do have an advantage because by tying this to the adenosine A2A receptor, it turns out that yes, A2A can modulate reward, but the A2A is really only truly expressed in one place in your entire brain. So spinal cord, it's upregulated pretty broadly, as I just showed you. In the brain, 97% of it is in one cell type, just one, in one place, the striatum. And so 97% of it are in dopamine 2 receptor expressing medium spiny neurons. So these D2 MSNs. And so just by being able to tie the terpene effect on reward to D2 MSN, to the adenosine A2 receptor, that allows us to tie it to the D2 MSN without really knowing anything more. At least gives us a strong premise, because almost all the receptor is just in this one specific location. Good for us, right? We got lucky on that one. Now, of course, that alone, that's an inference. That's not alone enough to establish the model. So this is going to be a major target of our future efforts to knock down the A2AR in the D2 MSNs, to do all the back and forth experiments, activation and inhibition, selective knockdown of D2 MSN, measuring activity, and so on, to demonstrate that it actually is through this site. But so far, just by knowing that, we can construct a model for how we think the terpene is modulating reward. So how through this reward modulation of opioids is occurring. And so that model is this now, of course, coming from the literature. You have the neurons projecting from the ventral tegmental area. They release dopamine. They release them onto D1 expressing MSNs and D2 MSNs. And so the D1 MSN is a little bit more straightforward. This is a stimulatory receptor, and this neuron population seems to promote reward, broadly speaking. It's very complicated. Please don't yell at me if you study this area. I know it's very complicated. This is dumbing it down. That's pretty straightforward. The D2 MSN is a little bit more complicated because the D2 MSN neuron population seems to block at least some aspects of reward and motivation. So this neuron activity alone can do that. The D2 is an inhibitory receptor, right? And so dopamine is released onto the D2. That promotes the inhibition. That reduces the activity of the D2 MSN, reducing this reward inhibition, thus promoting reward activity. And so the activity of dopamine on both of these neurons promotes reward. Just does it through a more complex mechanism. All right, so that, as if that wasn't complicated enough, now let's add the A2A into this situation. So other literature, not on terpenes, but on the A2A, shows that the A2A is inhibitory on the D2 receptor. It can heterodimerize with the D2R to directly block it. Also, it itself is a stimulatory receptor, so it can directly stimulate the activity of the D2 MSNs. And so both of these activities, stimulating the D2, blocking this break, taking away this dopamine blockade, is going to increase D2 MSN activity. That is going to tend to restore this break on reward and decrease reward. And so what we think is happening is that BCP, by activating the A2AR, disables the D2, increases the activity of the D2 MSNs, and blocks the reward that we see right here. And so just because we kind of got lucky, from these relatively few pieces of data, we can construct a fairly elaborate model for what we think is going on because the activity of this receptor is so restricted in your brain. But again, of course, we still need to prove that. And so Hager and Emily and other students in the lab chasing this down to really fully demonstrate that this is indeed what is occurring. And so those are incomplete stories, but they're kind of in progress and most exciting and the ones I want to talk about versus just showing you all the stuff that's been already published and you can go read on your own. But these are the key messages that we kind of have to date from our work on terpenes. Promising non-opioid pain therapies, that's quite clear. Good efficacy, low side effects. We're pursuing that. They have a lot of benefits to potentially developing. They may be a unique addiction prevention strategy, blocking the addictive effects of opioids as well as others. That could be used as a combination therapy in pain. It could also potentially be used just as a way to treat addiction on its own. We are working on both sort of approaches, and they seem to have a common mechanism in both of these areas of therapeutic effect through the adenosine A2a receptor, which was a bit of a surprise when we first started this off. We didn't think it was going to be through this target. I don't think this is the only target terpenes can activate. In fact, we know it's not. We've directly demonstrated they can activate at least three GPCRs. We know it doesn't activate the mu opioid receptor, but other than that, it seems to be pretty promiscuous, or they seem to be pretty promiscuous. But in these areas of therapeutics, it does seem to be through these receptor, and we're really enjoying chasing down these mechanisms of action and figuring out how this actually works, along with developing the therapeutics. And then lastly, these are easy to translate. So we can get these into humans pretty quickly with products that are actually on the market versus having to get raise $10 million in funding, do all the toxicity studies, blah, 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 all that effort that is required. And so these are the take homes. Oops. Wrong. These, of course, are the people that made it happen. This list is now too long to fit on one page, so I tried to highlight the people that directly contributed to the project and or are currently in the lab that contributed to this. So thank you to all of them. Thank you. I see Emily's online, so thank you to you, Emily, but to everyone else too. These are some of our collaborators that have worked with us on this particular project. The funding for this project has mostly come through the NCCIH. Thankfully, it didn't just get demolished by the Trump administration. That did not make it through the Senate and House. And then I do have a few conflicts of interest. So Napriva and Botanical both work on terpene products. So just know that I'm working with them to develop these products. And so keep that in mind when you look at my data. And so thank you to my lab. Thank you to all of you. And happy to have a larger discussion on this or on anything you want, really.
May I start? I would love to start.
Please.
Okay, so this was amazing. I loved hearing how terpenes affect pain. So I am not currently in a lab, but my goal over the next couple of years is to get a PhD in pharmacognosy, and I'm interested in in researching both discovering and then doing genetic modifications, kind of like what you're doing, and maybe a little bit different with stress and stuff, to look at both terpenes and any other secondary metabolites of plants to use against chronic inflammation. And one of the compounds that I want to study is actually limonene. So I was really happy to see
There you
that
go.
on there. And I've done a little bit of research on some of the other ones that you presented on, but not in the pain sector. And so the
Okay,
first
so you said pain. So I want to show you something. You should see this before you start cheating. This is not the last word. I'm not the only data that matters. But I have something to show you that could really change your future plans. All right, so hold on. All right, am I still screen sharing? I'm starting to
Yes,
lose track here.
yeah.
All right, so let me see where was that? I think that was in the blends paper, right? Yeah. Yes. Here is myrcene. Here's
Mhm.
D-limonene in CIPN.
And can you remind me, I don't do mouse work. Can you remind me what CIPN is?
Sorry, that's the chemotherapy-induced peripheral neuropathy. So that a lot of the data that I've shown you is in this model. Not all of it. So there's paw incision fibromyalgia. We try to do more than just one pain model. But at least, this isn't the last word, but at least in this one pain model, in this paper that we published, we found the effect of myrcene. I just showed you all that data with the other 5 that we usually study. But D-limonene so far is the only one we've tested that did not have an antinociceptive effect in CIPN or any other pain model.
Well, that's great for me because now I don't have to worry about you as a competitor. It does all these other cool things. Just to share a little bit, it will
Yeah.
reduce LDL-C if your LDL-C is high in humans. In I
Oh,
think
cool.
either mice or rats, they injected it into the gallbladder and it dissolved gallbladder stones. It cures heartburn in humans in like 2
Cool.
weeks, and it kills the
Oh,
bacteria
I
that
need
causes
some of that.
strep throat and also plaque. So like, I don't know, I'm doing a little bit different thing, but it's so cool to hear about other ones as well and the different
Yeah.
things that things that are they're fascinating. The fact that you can use them to modulate like reward receptors is a really, really, really cool idea. And I'm excited to hear what you're gonna do about that, what you're gonna do with that in the future as well.
Yeah, and there was actually a paper that worked on limonene and showed the benefits in anxiety. And actually, that one did that. The only other paper that I'm aware of did suggest that it was through the adenosine A2a receptor.
Cool.
Um, so some synergy there.
Thanks.
Hello, Dr. Streicher. It's your
Hey,
favorite
hey, good
yapper.
to talk to you again. How you been?
I wrote down notes so that I could try to ask my question in a concise way. So, whenever I was seeing your BCP, very beginning, when you were like giving indications that these terpenes have in different diseases, basically, obviously, if you remember from the classes that I've had with you, I am now a liver person. Still neurodegenerative, but I'm still definitely working with the liver. And I found it very interesting that BCP can help with liver fibrosis. So while you were talking, I did like a quick Google search.
That's
I
cool.
wanted to ask you, because of this and this idea of it being able to help with inflammation to the point that you're able to slow down the progression of liver fibrosis, do you think that this like adenosine A2a receptor activation is not only playing a role with lowering those type of microglial like communications with neutrophils and macrophages that kind of transcends past the CNS pathway into these like hepatic cellular Kupffer cells, which are like the immune system of the liver, by the way. Um,
Yeah.
but like I find it fascinating to think that something like this that has more activity in the brain than it does in other places is able to kind of stretch itself past that barrier. And I'm now like interested in wanting to go and search up using terpenes as like a co-therapy for something like hepatic injury or
Mhm.
fatty liver disease or something. But I wanted to hear your thoughts on what you think of this like microglial communication between the brain versus the liver, like the liver of all things, you know?
Yeah. Well, I don't know a lot about the liver, but I think it's definitely possible. So let me pull this up here. So this is some of the data that Jerry did. I think he should have. Yes. Yeah, so we're getting like what would that be? Probably about a little around 40% colocalization with IBA1 in the A2A. And so
Mhm.
this oh yeah, that's a nice one, right? So you can see like that A2A showing up pretty nicely here in that microglial cell. Um, yeah, this one showing up there too. And so we definitely see colocalization of the A2A in the spinal cord, especially in that CIPN state where it gets upregulated. We haven't really focused on that, but yeah, I think it's possible. One potential challenge, though, could be that the A2A is a G-alpha S stimulated receptor, and thus, just first pass, you would expect it to promote uh, inflammation in the microglia. I haven't looked in the literature though to see what it might be doing in there. Maybe someone's already studied that. So there could be a way by which that would be anti-inflammatory. But anyway, I'm rambling a bit, but that, yes, we do see the receptor in microglia. I think there could be some possibility there. I don't, I don't recall having seen anyone in the literature figure out a mechanism by which terpenes are anti-inflammatory, simply that there are many papers showing that they are.
Yeah.
So yeah, there's definitely could be something there. I just haven't looked enough to say what is or is not known about the mechanism.
No, that's completely fine and valid, honestly.
I think
I
Carolyn's
mean,
got an answer for us.
okay.
I do. I'm sorry. They're ROS inhibitors. So the structures are very like they're kind of I forget the name for the chemical structure that they're kind of like quinolones where they've got the conjugated bonds or there's something like that. So they absorb like ROS damage and then make it go away. And there's other
Nice.
mechanisms too. There's like three or four and then they all come together, but that's the one I remember.
So at least that one then sounds like it's not even protein mediated. It's just like a direct
Yeah,
interaction.
that's insane. Yeah, I'm thinking about like the full body effect of something like this for it to be not only pain management in the nervous system, but also like because if you think about it, your Kupffer cells are helping with damage of the liver, which even though we're not getting the pain signals from the damage being done, they are also being activated through this system and are causing these type of therapeutic-esque effects. So I like it. It also kind of plays into the role of biological relevance of this type of receptor action.
Yeah.
Being able to treat things that you're not necessarily feeling pain wise, and recoding that into something else, which I like. Very interesting.
Oh, I did think of this too. So we haven't followed up on this, but we did show that all five of the ones that we tested very nicely activate the CB2 receptor, and that has been tied to inflammatory regulation,
Mm-hmm.
as in anti-inflammatory, quite a bit, right? In peripheral
Yeah.
cells, I don't know about Kupffer cells, but definitely in the periphery. And so that could be a mechanism by which they would directly be anti-inflammatory, as well as the potentially like the ROS scavenging or the other things that have been mentioned. But these are dirty drugs. I say that non-derogatorily. So, like just in our hands, we show they activate the CB1, the CB2, the adenosine A2a receptor. Others have shown activation of like TRP channels, like those not retinoic acid, but one of those like fatty acid hormone receptor type ones, not like estrogen receptor, nothing that simple, but like RXR or something like I forget which one.
Yeah, RXR, LXR. I study liver X receptor because I'm the liver person. So, yeah.
Yeah, so there are a lot of potential targets, and then maybe just like those direct chemical effects, right? Like Carolyn was just saying, that's really cool. I hadn't actually known that one before, and so they do a lot of things, and in one sense that makes our jobs challenging, and it does kind of, I guess, cut against this. I don't know what you call it, dogma, received wisdom in the pharmacology field that we should try to make drugs that are as selective as possible. And so anytime you have something that's dirty, people like the reviewers immediately start like, but actually a lot of these natural product drugs that do have a beneficial profile, many of them are very dirty. So terpenes, we demonstrate they've been tested in humans. They're generally non-toxic with low side effects, potentially very beneficial, and they're dirty as hell. Another example would be something like buprenorphine. There's like at least eight or nine receptor targets this thing hits, but it has a better profile in terms of addiction potential and so on than a pure opioid agonist. So dirty is not necessarily a bad thing. It does make it challenging to study, though.
So I had a mechanistic follow-up question. When you were saying that the A2A can heterodimerize with the dopamine 2 receptor, is that actually blocking activity or is that inducing a different signal transduction cascade?
No, that's a direct interaction, and that direct interaction directly decreases the affinity of the D2 for its ligand. And so they've shown that using like binding assays, for instance. And so there's a direct physical interaction that at least decreases that. There could also be like downstream receptor, like cascade interactions, but at least part of the explanation is like a direct heterodimer that's the H2A is actually inhibitory on the activity of the D2.
Okay, awesome. And then my other question was, have you seen any activity with this cohort of terpenes on the kappa opioid receptor?
Haven't tried.
Okay.
That would be a good question. We have tried the mu, and it does not activate the mu, at least in vitro.
Okay.
We haven't really done anything in vivo, but at least in vitro, it does not activate the mu. That doesn't mean it doesn't activate the kappa.
Yeah.
Um, you know,
My guess is that
Obviously
the
these are
ones that
pretty
are
dirty.
aversive would have at least some activity there. But
Yeah, that's a good thought, right? That could be a mechanism by which it's doing that. At some point, I'd like to be able to do like a panel screen, like send this to SERAP or psychoactive drug screening program or something like that, and get a cleaner idea about what these things are activating and what they're not.
mhm.
I mean, some of our reviewers have suggested that this could be like a non-receptor mediated mechanism, like it's interacting with, say, the membrane and changing the fluidity, and we know that alters GPCR activity. I guess that's still possible, but at least some of our data suggests that we do get a direct interaction, and that we don't activate the mu. So it's not like every receptor. So there's at least some reason to believe that it's an actual ligand-based effect.
That's interesting. I was also wondering about with this interest in looking at terpenes for depression, how are you going to induce depression?
Yeah, so the one that Emily, well, maybe I should let her speak for herself, but the one Emily wants to do is this, oh, what's the acronym? Okay, Emily, you better chime in. I can't remember the acronym. It's something,
It's called UCMS.
that's right. Unpredictable chronic mild stress. There we go. And so it has all these different stressors, like, a wet cage, no bedding, tilted cage. I think isolation was one of them, right? There's like these different stressors, and they get a random stressor every day for 2 weeks. And so they don't know what's coming. It's not like the end of the world, but it just sucks and it's ongoing and it's unpredictable. And that induces anhedonia and depression, like decreases their sucrose preference, induces depressive behaviors. And then Emily's going to combine that model with the terpene treatment and see if it restores them. So we're like 2 bottle choice. We have a automated cage monitoring system we're going to try to use for this. Some different approaches to try to get at that. One of the things she found, though, is that like some of the traditional assays, like a tail suspension test, there was a direct confounding effect because we already knew that these terpenes change motor activity and they're sedative, and so giving them a terpene right before you do the tail suspension test increases their immobility time. And so it looks like a pro-depressive effect, but that's a confound. So we got to find ways to get around that and to use models that are probably more translationally valid than something like forced swim-induced despair, which honestly also makes me feel like a bad human to use that on the mice.
May I ask one more question?
Of course.
Well, have you thought of using a microarray to look at which genes these different terpenes turn on or
Yeah,
affect?
I mean, we haven't got to that point, but that would be great. Or proteomics. Like, so we like to do proteomics with the Paul Anglis here down the hall. So yeah, getting a clean idea about what's happening. I think what I'd like to do would probably be not to do tissue based, but to try to get at like specific cell types, right? Because this is dirty enough that if we just do tissue based, it's just going to be an amalgamation of all the different things that it's doing. So we need a little bit more selectivity. So I don't know, maybe PAX2 neurons, for instance. Single cell proteomics, single cell RNA-seq with a marker for PAX2, something like that, and show the efflux. That would be cool to do. Right now, we're a little tight on funding, so hopefully we can get more funding to support these major studies, and then yeah, like we could explore some more interesting things like that.
Yeah, that'd be great. I really loved like all the mechanistic stuff you're doing. I did before I worked with Dr. Thatcher, I worked for Dr. Zhang in the plant sciences department doing virology research for about 3 years, and we did a
Mhm.
lot of genetic engineering and plasmid creation and constructs and stuff
Yeah.
like that. So I like all the reminiscent stuff of that. It's given me ideas for my future. So thanks.
Well, that's great. Initially, I trained as a cell biologist, so it's like I'm doing all this behavior now, but I still have that stuck in me somewhere.
It's fun. Cell culture work is fun, and like all the imaging and all the different things you can do with it, and it's like an endless possibility.
Yeah, of course.
Just
But
doesn't
one of the
trans.
things that we're talking about in the department is trying to get more of these like this is hard in neuroscience, but trying to get some more of these in vitro models of neuronal function, like organoids, iPSCs. That's fairly established at this point, even for neurons. But like organoids, trying to reconstruct circuits. There have been some really interesting papers lately of people sort of using organoid and in vitro approaches to actually reconstruct like circuit pathways, and then seeing how those pathways are modified. So along those lines, something like that would be really cool. And then, of course, combining that with terpenes or whatever to be able to monitor activity changes.
Yeah, awesome. Well, I'll keep an eye on seeing on what you guys publish, and maybe we'll hope to have you back and have another talk at some point.
I have another question. So when I think about the D2 receptors, my brain immediately goes to prolactin.
Hmm.
And of course, we already have work on that in the department. And I'm wondering, have you looked at the expression of tyrosine hydroxylase containing neurons in the hypothalamus in CIPN and with morphine and terpenes, and whether that expression is changing?
Hmm. Yeah, no, we haven't, but that's an interesting idea. The only one we've looked at so far is the VTA, and that was a pretty simple experiment, really, just the conditioning with the different treatments, right? And so those different areas of the brain, I know that pain changes the reward circuit, right, and alters that. Maybe terpenes restore that. I don't know. That would be really interesting. Like so far, all the reward studies we've done have just been with like uninjured animals, but we know that the pain changes that. So combining those would be great. I put in an RO1 recently, the reviewer to study the reward stuff. The reviewers unfortunately were idiots, but we're going to resubmit this turn or this round, and so hopefully, like getting something like that funded would give us the space then to look at some of those additional questions, which are highly relevant.
Yeah. Yeah, I'd be really interested in seeing if we have a different, like a change at the organizational level of that inhibitory mechanism rather than just more dopamine or less dopamine. I think that would be really
Right.
interesting to see.
Well, and we know that happens in pain, right? So we know pain changes those circuits. So the question then would just be, terpenes, does it restore them, or even does it change those reward changes in pain? We don't know that yet. And if it did, would it be because of the pain relief or sort of a direct effect on the reward circuitry? Both could be possible. So a lot to dig into there.
Have you also, with your work, seen sex effects? I saw in some figures that you had them labeled male and female mice, but it wasn't labeled all the way through, so I wasn't sure how much of it was just males or females.
Right. Yeah, I pulled those from different papers, so sometimes they're labeled differently. All of our work is done with males and females. I cannot think of a single sex difference we found so far for terpenes. Um, okay, no, that's not entirely true. There was one. So I don't know why it happened in the blends and not the individual purified terpenes. But we had this paper. So like all the work, almost all the work I showed you, or all the work I showed you was all in isolated single terpenes. It's like figure out what they do before we start getting more complicated. But of course, the way that most people actually consume these is extracts, and those are complex extracts. And so we got some extracts from these folks here at Terpene Belt Farms. Omar El-Sisi just visited our department a little while ago, and we actually did see a sex effect there. Yeah, so like comparing the effect on males versus females of these three different blends. And it was much less effective in the females. You look here at the AUC versus here, much less effective in the females than the males. We haven't seen that in our single terpene work, and I don't know why. But in general, we've found very few sex effects, just this one. And then when in that case, we always run males and females, don't see a difference. We combine them. So all that data I showed you were combined groups of males and females.
That's really interesting. To me, that indicates that we have a sex hormone nuclear receptor mechanism going on. And if it is an allosteric effect, it would be really interesting if there's some sort of synergistic allosteric effect that only acts on certain hormone receptors.
Yeah. And we did show, I believe, yes, we did show that you could block all of this with the adenosine A2a receptor antagonist. So you could still block it all. But I don't know exactly what's going on, because these mixtures are quite a bit more complicated than of course the purified single ones, right? So these are the three ones we used. And as you sort of look at the different symbols and the different levels, we didn't even include anything under 1%. There were several minor components under 1%. You see there's quite a complex mixture of different molecules in here. So I don't know where that effect is coming from.
Does the A2A receptor have downstream signaling effects on hormonal signaling?
I don't know. Good question. My guess would be yes. That's a very big. Okay, you're saying yes, Carolyn. Sounds like you know better than me.
No, no, I said good question. No, I said good
Oh,
question. No, I have no idea.
I think I don't know, but I think it's very plausible because the adenosine system in your brain is very widespread. And like the adenosine is kind of like this ongoing modulatory signal. There's a lot of it. It's kind of being released all the time. And the adenosine is in a lot of places. And now we're talking about other receptors in the A2A, because the A2A is more restricted in location. But of course, there's other adenosine receptors. So I'm sure that there's downstream interactions with neurohormones.
Yeah, I think that your evidence that the A2A receptor is like that funnel point, that part is really clear. So the question is what happens afterwards that would elicit that
Yeah.
sex difference. Um, but yeah, that does seem kind of complex to parse out.
That is, that's the unfortunate thing, right? Once you start working with these complicated blends, something that a consumer would actually consume, right? So this is a lot of what you will find in a dispensary, like extracts, crude. I mean, not crude in the sense that they're like got ethanol or whatever in them, but just sort of like not fractionated. There's sort of similar to the original composition of the extract, and I go into a dispensary and it's kind of interesting, right? So they're like, oh yeah, you take this thing, it's got beta myrcene. This is going to make you feel this way, and this other is mixed with linalool. That's going to make you feel this way. So they're like making all these claims, but there's, as far as I'm aware, not any controlled data on any of that. And so part of my motivation is to sort of chase the commercial space and get some actual information out there for people. I think they mean well. Like I don't not accusing them of anything, but they're making a lot of claims that are not, I don't think, very well backed. And like as a great example, this company Leafly, they really focus on selling terpenes. They have like this giant wheel diagram with all the different terpenes, and each one says how it's going to make you feel, like creative, fresh, awake, like calm, like all these different feelings that it's going to make you feel. I don't think there's a lot of reason to think that.
Yeah, I have some friends who are really into it, and I've definitely had conversations with them before where it was like, okay, you have read many papers, you have done a lot to educate yourself, and you're wrong. The data does not support that claim. And I think a big part of it is this assumption that any paper relevant to terpenes, any paper relevant to cannabis is going to be widely applicable, when there's actually so much diversity to the chemical milieus that that's an
Oh, yeah.
absurd thing to truly believe.
Yeah. And I mean, and some of the companies are trying to get around that, right? So like when Omar was just here like 2 weeks ago or whatever, like, okay, the CBD THC 1-to-1, that's what we want. So we're going to like use genetics and design a plant, which is going to give us that. I'm like, okay, that does sound cool. But on the other hand, why can't we just make a product that combines those in exactly that amount from purified precursors? Maybe I'm being too literal about this, but I guess there's a place for both.
I like the idea. I think that's way more practical. If you can make it and it's still mostly natural, like it's coming from a natural product rather than being synthesized, and it still has all its post-translational modifications like it were a plant, and you don't growing a plant takes forever, and you never know what you're
Yeah,
gonna get either. Like the compounds in it can change, so it's hard to standardize that.
it really is. Yeah.
Do you
And
guys
that,
do
you see that, right? So like even again, looking at this graph right here of the different terpene extracts, these are the same plant at the same place. Like these aren't even different locations, right? This is the same field. And they kind of grow these different batches, and then you get these different, was it sunny the week before they they did the harvest? I don't know, like moisture content in the soil, like all these things can really change that. Hard to standardize, as you say. And so,
Yeah.
as a pharmacologist and someone designing therapeutics, I would rather just make a product from preset precursors. You know what the concentration is. QA is much easier. Most of the companies I'm working with are kind of pursuing that kind of approach.
It's practical. It's very practical. I'm not like I'm interested in like I love the idea of just asking questions and finding answers. If I wanted to make a product, I would do the same thing you're doing. I was going to ask you, do you guys do fractionation in your lab?
Kind of. So, like, Chris has been helping us do this a little bit recently, in terms of being able to like fractionate extracts from tissue. We're like doing screening, like drug screening. We haven't done anything from like plants. I guess in principle we could. Like, where Chris is getting the HPLC with the fraction collector and all that set up.
Cool. That's one of the techniques I want to learn in graduate school. That's what I'm like, I mean, when I want to learn, and then I'll do some CRISPR too, I'm sure, and all that kind of stuff.
Sounds like you got a plan. That's awesome.
Yeah, I do. And I'm still leaving it a little open for leeway because I've got a little bit of time. So that way if things change or, if I find like a PI that I really, really, really want to work with, I've got some people in mind, but I haven't talked to them yet. So who knows how that's going to go.
All right. Well, I hope you'll consider us. So ABBS is always taking applications.
I would love to. I don't have anybody in Tucson that I'm considering. So that would be
Oh,
very convenient for
fair
my family.
enough.
So thank
Caroline,
you.
you need to stay. Stay here.
The
I'm trying
PhD
to
that
help you, Sophia.
get costs 20 grand. So there is a financial factor in that I could go to school for free. So I want to go so my goal right now, Dr. Streicher, is I want to go check out Cartagena, Colombia this year and see
Oh, wow.
the University of Cartagena. I'm bilingual in Spanish. See if their
Well,
university, where it's laying on the science scale, like, are we backwoods? Do we have equipment? Do we have, NMR and HPLC and all of that? And if it's set up, I want to get a PhD there, and I'm really interested in doing like bioprospecting and collecting plants from the Amazon and testing them. And it seems like it'd be really easy to do that when it's right next to you. But that's a $20,000 degree, and they don't guarantee in Colombia, they don't give you a stipend to live. So like I have to raise money for that or decide to get a degree in my country and live a nice, easy life. So yeah, so there is definitely leeway for staying here. So that sounds great. Thank you for the
well,
opportunity, the thought.
there are there are places that are kind of doing what you talk about. So I can get you the name if you're interested, but like Chris and I were communicating with this group that has like a biorepository of plant extract, other extracts too, but like plant and fungi. And so they've got like thousands of these like extracts, archived and so on that you can get to screen against targets of interest. We have some of them in our freezer actually.
Very
Um,
cool.
so there
Yeah,
is some of that. And also
def
opportunities to like get a degree here, but then go to those places to do that. And so as an example, Chris and I, we're intending to set up this, it's almost ready, this like microfluidic platform for screening bacterial species. And one of the things we want to do is go collect sponges from like Puerto Penasco or Hawaii or other places, encapsulate those bacteria, then basically bioprospect those to see what interesting things we can find.
there's so much cool stuff. Like I've got this idea. They're doing bioengineering for so many different things, but you can bioengineer bacteria to produce anything for you, basically super
Yeah.
easily. They're so easy to grow, and you just don't tell the people that they were made in bacteria because nobody wants to hear that, as long as you purify it right. So yeah, there's so many cool biotech, like bioengineering things and ways we can go with all the different
You
stuff.
should talk to Chris. So he actually does exactly what you're talking about. So like isolating gene clusters to do semisynthesis or modified bioproduction, like that's a lot of what he's doing. Screening, production, finding natural products,
What's his
bioprospecting.
last name?
Cartmel, Cartmell.
Hartmel. Perfect. I will send him an email for my personal email saying that you said I should reach out and we should talk about science.
Caroline, if you'd
Absolutely.
like, I can introduce you.
That sounds great.
Okay. Yeah. He's done tons of really cool natural products stuff.
Yeah, he's helping Sophia actually with her BPA measurements.
Yep.
How's that going, by the way?
Um, it is going, and so it's going
Long
well.
pause.
Tomorrow we're gonna do the LCMS prep for serum, and
Okay.
we'll send that in. And essentially, as of our current plan, I will be doing the prep and then, using LCMS to observe how much BP there is for like 7 different regions in the brain for
Wow.
each of my animals, and then also serum. And so I have some dread about the process of optimizing for all those different brain tissues that are all just a little different from each other.
Hmm.
But I'm sure it'll end up being okay. And tomorrow we'll do the easiest, which is just serum, which we've done before.
Yeah, I would. My only advice would be to get some like animals that you don't mind losing, and
Mhm.
just do the optimization on those before you do your precious samples. But I'm
Yeah,
sure
I
you know that
have
already.
about 3,000 tubes
Dang.
that I could throw away tomorrow.
I wonder if too if there's a way to, if you're dealing with large sample numbers, if there's some way to automate or at least partially automate the preparation and handling, right? So I know there's big handling robot down in functional genomics. Like I wonder if there's a way to speed that up.
Yeah, quite possibly. On the bright side, the ones that I have to prep right now, it's like 40 animals. It's not a big deal. So I can just have 1 day per tissue and just have, 7 or 8 really long days. And
Yeah, I was about to say that's like 280 samples, but then yeah, if you can break it up over the days, and then yeah, that shouldn't be a problem because each region will have its own internal controls. So it doesn't matter that you did different regions at different days or analyze them at different times as long as you have your controls in each set.
yeah, it's just about comparing it. And this is with the dose response study,
Hmm.
because essentially we have this question of, okay, we're seeing the molecular changes in, the PAG or the RVM or the VC or wherever. Is that actually because there's BPA there, or is it downstream of something else? And so
Hmm.
if we can actually link that the animals that have higher BPA in that brain structure also have this effect, I
Right.
think that'll be more substantive. So that's the approach we're taking right now. But I've got a bit of a headache over the whole periorbital allodynia nightmare. So
What nightmare is that?
that would be that all of the animals I got this summer had periorbital allodynia on baseline. Yeah. And so that was a lot. And some of them, ended up having a high enough threshold that I could work with them. But a lot of them were just anxious wrecks all summer long. And
Wow.
so it's pretty difficult to parse. And they were like MMA fighters. Like they would see the filament coming and they were bobbing and weaving and whacking at it. And it was just more than I've seen with any of the rats in the past. It was absurd.
Oh man, I'm sorry.
It's all right. I have other data, luckily, but yeah,
It's a tough go.
yeah.
All of them. It makes me wonder if there's some kind of transport issue or like people are
Well,
banging on the walls down in the UAC or something.
there's new bedding.
Ah,
Yes.
yeah.
Yeah.
They don't seem to think too much of like changing the food, changing the bedding, whatever.
Yeah, it's insane. Especially because with the food, there's phytoestrogens in that, so that could dramatically change one of my studies.
Right?
They did that. But yeah, anyway, overall, it's pretty good. And from my conversation with Tally this morning, I'm hoping to get 2 papers out of the animals that I worked with this summer. So
Okay.
it's good.
That's that's pretty damn good.
Yeah. So hopefully we'll make it
Yeah.
happen. But anyway, I wanted to say thank you so much for coming and talking with us. This was a really good talk, and I was really happy to see that there were a bunch of people who came.
Yeah.
And yeah, I'm really happy that you're helping us to get this off the ground.
Of
Yes,
course.
thank
Let me
you
know if you
so
need any
much,
help.
Dr. Streicher.
Yeah, thanks. Let me know if you need anything else. I could try to help you recruit people to speak or
I would love that.
publicize it. I don't know, whatever. Um, just let me know.
Yeah, no, I'd
And then, Carolyn, if you want to
absolutely
talk
love
to
that.
Chris or follow up on our projects or talk about natural products or apply to grad school, let me know.
That was great. Yeah, I will I'll take a closer look at what you guys have been doing and I will keep that. It's a really good thing for me to keep in mind. And a very pragmatic thing to keep in mind as well. Even though I don't pragmatism isn't my favorite thing, but I
What?
do.
No.
Well, spending that time in Cartagena sounds pretty cool, but it's
I have
obviously you got to figure it out.
I have free miles on a credit card, so I'm gonna take whenever I forgot a
Little
good
trip
time,
down
I'll
there.
spend a week or two down there and like the U's right downtown, and so I'll do that and check it out. But yeah, it would be so much more financially beneficial and just there's a prestige to getting a degree in the United States. I've
Yeah,
known to myself, I'm like, if I go get a PhD in Colombia, I can't come back to the US and work with that degree ever. And everybody loves a PhD from America. And so
yeah.
there's a lot of benefits to doing it the other way around. And I didn't see anybody at the U of A who was doing anything close to what I wanted to do. Honestly. And so the fact that there might be somebody closer to like what I want to do makes it a lot more exciting.
So,
Right.
Caroline, one thing you should consider is that there's a model where you could be getting the PhD here, but going and getting samples there.
Yeah,
And my
exactly.
dad has multiple PhD students right now that he is like, they're from Ecuador and he sends them to Ecuador and they go and they get their samples and then they come back and they do the data analysis with the fancy equipment here using samples from there.
That would be like ideal because there's no way the equipment's going to be what I'm looking for. There's no way.
Mhm.
I don't even know what I'm going to look for. I will look at that, but that would be awesome. Yeah, we should talk about that sometime, or I should talk to your dad about it if he's around sometime. If
Yeah,
they're
he
on the
might be able to give you ideas for people who would be open to setting up a project like that or who have projects like that. I have no idea who's doing that stuff, but
Cool.
there is a model to doing a PhD where you get to travel to cool places.
That would be so awesome. Yeah, because it's like there's the best biodiversity, the most biodiversity in the world is in the Amazon. And so I love the idea of going out and looking for things that people haven't looked for before. And there's not too many white people who go down there and pick up plants and bring them to other places and stuff like that.
Yeah.
If it would help, I mean, you may not need it, you may already know, but if it would help, I have a former student who's currently a postdoc at Texas, and she's from Colombia, and she may have some advice on the school situation there.
Ooh, yeah. I would
She did her PhD here, obviously, but in my lab, but may know something about the system.
so she's a postdoc now in your lab and got her PhD in Columbia or the other way
No,
around?
no, she got her PhD with me, but she's from Colombia, like moved here after her childhood.
Cool. Yeah. I will definitely reach out so you have my email, Dr. Stryker, because I'm not at the U of A anymore, so I don't have a U of A email. I have it, but it doesn't really work. It's got
Yeah,
some
just
weird.
drop me a line.
Yeah, and then we can chat a little bit more. And yeah, because like I don't have none of my friends who I have one friend from Columbia, but she never went to college, and so like it's hard to ask her questions about that. Beautiful.
Well,
Thank
she
you.
went to college here too, so maybe she might not know enough with specificity. I don't know.
Who knows? It's always good to talk to people who are from someplace, and they can always give you some insight or say yes, I like it,
Yeah,
or give you
absolutely.
tell you to watch out for something. So you can always learn something. So thanks.
Yeah. And then
Awesome.
I'm co-director of ABBS. So if you have any questions on that, like applying, if you do want to come to the program, then I'm your guy to answer on that too.
Oh, awesome. And then ABBS, what is it? Will you tell me what the acronym is? Because there's so many in the
Yeah,
pharmacology department. Is that
it's
what?
the Arizona Biological and Biomedical Sciences graduate program. So this is an umbrella program that covers 12 or 13 different programs. And so like plant sciences is one of them.
Okay.
Medical pharmacology, our program's another. Pharmacy, the different pharmacy programs. Molecular and cellular biology, immunobiology. So it's like a bunch. It's the largest umbrella program on campus. Um,
Yeah.
it does not include chemistry, biochemistry, which could be another potential interest to you, but I don't know how many of them are really doing the kind of stuff you're interested in. And their graduate student situation is not as ideal.
Yeah, Caroline and I have enough of the chemistry department for life. We're good.
They're nice, but yeah. So when I moved over to North Speedway, I'm like, North Speedway has all the
Oh
money
yeah,
for all the resources, all the research. Like, I love working on an R01 grant and all the different things we
yeah.
get to do. And everybody in Dr. Thatcher's lab, except for maybe one person, is in ABDS. So
Okay,
I
yeah,
the word before, but I was like, which one is that?
yeah,
So awesome, cool. Well, what a fortuitous day for me today. Great, thank you guys so much. And I
absolutely.
will let you guys have a great rest of your evening with that.
Good
All right,
night.
guys. Talk
Bye.
to you later. Thank
Thank
you.
you.
Thank you guys.
Good night.
Night.